So, welcome back. We're on to intercellular junctions in plants and animal cells, but primarily animal cells. So here we are. Between cells are junctions that connect them in various ways and enable them to function together sometimes as well. In plants, we have plasmodesmata. We looked at those already, but those are basically gaps in the cell wall and the membrane of two adjacent plant cells so that you can get materials passing from one cell to another. Animal junctions look like this. There are three types. Tight junctions are junctions that seal adjacent epithelial or lining cells together, and they are made up of proteins in the plasma membrane of adjacent cells that physically contact each other. And what they do is, there's several functions. So here's a nice example of the tight junction here between the cells that line the small intestine. And so you can probably recognize these cells as having these little projections called villi on top. Those are the cells that interact with the inside of the small intestine, right? And between them are tight junctions that seal these two cells together. Two functions of tight junctions include they prevent the passage of materials between the cells, so nothing can get between the cells. Anything that wants to pass from, say, the lumen of the small intestine into the tissues of the body has to go through the cell. The second thing that they do is they prevent the cell from changing polarity. So this particular cell has little villi, little projections that sit at the lumen of the small intestine, the inside of the small intestine. We don't want that cell to do a 180-degree turn and have its villi in the wrong direction. The tight junctions prevent cells from flip-flopping, if you will. Okay, gap junctions down here connect the cytoplasm of adjacent cells. What these are essentially are little gaps or passageways from one cell's cytoplasm to the cytoplasm of another, made up of proteins that can open and close. These allow materials like ions and nutrients and hormones to pass from cell to cell, very similar to plasmodesmata. And then there are desmosomes, right? Desmosomes up here are localized patches that hold or rivet together two cells so that they're secure. I guess that's the best way to put it. What they're made up of is, you can see these little red stringy things. Those little red stringy things are keratin proteins extending into the cytoplasm of adjacent cells. What these patches of keratin do, or these desmosomes, is they rivet together two adjacent cells, keeping them together so that they don't fall apart. Epithelial cells, cells lining things, like our epithelial cells of our skin, have a lot of desmosomes between them. That prevents the cells from falling apart. That's pretty important in our skin. Our skin gets a lot of mechanical stress. So we see desmosomes where cells have a lot of mechanical stress. This is an interesting fact. When I was reading up about desmosomes, I found out that carcinomas, which are cancers of epithelial cells, don't have desmosomes typically. So cells that are carcinomas don't have the desmosomes anymore. What that means is they probably can detach from the rest of the cells, and when they detach, they can move to another place. They can metastasize, like cancer cells sometimes do. Okay, so different kinds of junctions. The plasmodesmata and the gap junctions are more similar from plants to animals, but these other junctions are important too. Now for review. There are lots and lots of ways to review this chapter. A lot of important material in this chapter, so take your time, review what the structure of each part of the cell is and how it functions and how it relates to the bigger cell. That's it for now. Thanks.